Hi, thank you for having me. My name is Donna Evon, and my talk today, I'm going to be talking about giving kind of the broad background behind the PSC PROM project that we're going to be starting soon. And I'm going to be talking about PRO measures that have been used in the PSC literature historically. So just a little bit about terminology. What is a patient-reported outcome? So the definition comes from the Guidance for Industry that was published by the FDA in 2009. And this definition is that a PRO is any report of the status of a patient's health condition that comes directly from the patient without interpretation of the patient's response by a clinician or anyone else. And so the operative term here is that it comes directly from the patient. A little bit more about PRO terminology to help the audience. So think about a patient-reported outcome as the concept, the domain, or the outcome of interest for a study. And I've listed several here that would be relevant to PSC. And then think about the PRO measure, and this can be used interchangeably with it's an instrument, tool, or survey. This is the means by which we're going to collect data about the PRO concept. And it doesn't just include the questions on the survey, but it also includes the instructions, the recall period, and documentation to support the measure's validity. And then the PRO measure is taken by participants and it produces a score. And that score represents the PRO concept at a given point in time. And then in a PRO claim in the field of drug development, is when PRO measures are used in clinical trials to evaluate treatment benefits and risks directly from the patient's perspective that can be used later to support drug labeling claims for advertising. And so I'll use abdominal pain as an example. Let's say we want to study abdominal pain in patients with PSC. We might decide to use the PROMIS belly pain survey. We'll have several study participants respond to the survey and obtain a total abdominal pain score. We can then take that score and aggregate it with other patient scores and look at how those scores might change before, during, or after a treatment. And then if it does show that there's a significant change in that score, the pharmaceutical company is able to say that drug X decreased participants' abdominal pain by 50%. So why are PROs important in clinical trials? Well, if you take a look at this cartoon on the left, the doctor walks in and says, congratulations, your ALP level is normal. And the patient, though, might be having a very different experience. They're thinking, my PSC affects my whole life. I can't sleep. I'm exhausted. So this discrepancy is actually fairly common in the literature. There's a discrepancy between physician assessment and patient assessments of health status and what a clinical benefit means to healthcare providers and patients. So a clinical benefit is defined as a positive, clinically meaningful effect of an intervention on how a patient survives, feels, or functions. How a patient feels or functions with a disease or on a treatment, this is a subjective experience, and the best source of gathering that information is going to be from direct patient reporting. Treatments for PSC should impact underlying disease or impact how a patient feels or functions. Ideally, both. Ideally, we're looking for drug regimens, maybe multiple drug regimens that ideally work on both disease and symptoms. So this issue, this kind of notion of trying to go after disease endpoints as well as symptom endpoints was really echoed in PSC Partners' patient-focused drug development meeting with the FDA a couple years ago. On the left, you can see that they list these symptoms and daily impacts of PSC. These are really often what matter most to patients. And you can see this list here. These are patient-reportable concepts, domains, symptoms, and these are going to be best evaluated with PRO measures. And so we have to develop good measures that can be put into clinical trials so that we can evaluate the effectiveness of treatments on PSC symptoms. Now I want to take a step backwards. I'm going to talk about a review article here. Let's talk about what kind of patient-reportable concepts have been found in the PSC literature to date, and then what kind of PRO instruments have been used to measure those concepts. So we did a review study where we reviewed all studies from PSC from 1999 to 2019. And we looked at where there were studies that mentioned a Piero concept like quality of life or fatigue, or an instrument in the study design or results section of empirical scientific studies. So we excluded reviews, reports, and editorials. And you can see here, this table shows you the most common, frequently cited concepts, domains, outcomes. Symptoms was by far the most frequently cited concept in the study design and in the results. And then there was also several studies that looked at Pureitis, fatigue, abdominal pain, and then you could see below there were studies that looked at 19% at quality of life and functioning. So then the question was, all right, we have all of these concepts that are being looked at, evaluated in the PSC literature. How are they measuring these patient reportable concepts? And the number of studies that actually used a Piero instrument was only 52%. And so what that tells us is that the other 48% were using information from patients' medical records or judged by the clinician, but not information that was coming directly from the patients. This is a list of the most commonly used Piero instruments in PSC studies to date. And I've highlighted here a number that we described as non-specific rating scales, visual analog, or diaries. And what this means is that in those articles, there was barely any information about the instrument. There was no citation, no reliability or validity measures or information, and no copy of the measure put into the appendix. So these are Piero measures that are not going to meet FDA standards for a fit for purpose Piero measure. They weren't developed in patients with PSC, they were developed in other patient populations, and they provided very minimal evaluation of the reliability and validity for use in PSC populations. If you take a construct like itch, for example, how many studies that described or reported on itch used an instrument? No instruments were used in 56% of these studies. Again, when they talked about itch, it was retrieved from medical records or judged by the clinician. Two instruments, the itch visual analog scale was used 15% of the time. And this is an example of a visual analog scale. It's a line, and simply the patient just marks an X on the line to indicate their worst imaginable itch. Or a numeric rating scale like this, where a patient marks a number 0 to 10 to indicate their itch. And then if you take another symptom like fatigue, that's very pervasive in patients with PSC, again, only 50% of studies that talked about fatigue used an instrument to evaluate that. The rest of it came from medical records or judged by the clinician. When a Piero instrument was used, studies mostly used this fatigue impact scale. It's an old instrument. It was not developed in patients with PSC. It's long. It does evaluate three different types of fatigue, and it's been modified and it's been used a lot, but it's not been reliably evaluated in patients with PSC. The other types of instruments for fatigue, again, are visual analog or numeric rating scale, kind of like the ones indicated here. So just to summarize the findings of this review, Piero concepts were measured using a Piero instrument only half of the time, and the majority of instruments that were used were poorly described, non-empirically supported, one-item visual analog or numeric rating scales, and they lack details about the instrument's development. Now, there are 3 new PSC Pro instruments that have been recently published. I want to review those. The first one was funded by Gilead to use in Gilead treatment trials. It's called the PSC Pro, and it has 42 items. Module 1, which is shown here, is of 12 symptoms, and Module 2 is the impact of disease on life domains. So a patient would take each of these 12 symptoms. It's scored 0, no symptoms, to 10, as bad as you can imagine. Each of these symptoms, these 12 symptoms, are then summed into one total symptom score, and that's what we would be evaluating in a clinical trial. So one of the disadvantages of putting all of these symptom scores into one total symptom score is that it's a multi-domain concept. It's multiple symptoms wrapped up into one score. So one of the issues from the FDA is that if improvement in a score for a multi-domain concept is driven by a single responsive item, say fatigue improves dramatically, but other items like itch or abdominal pain do not improve, a general claim that symptoms improved can't be supported by this measure. So that could prove a little bit of difficulty with the use of this for symptoms. The next one is a 4-item instrument on cholestatic complaint score. And you can see the 4 symptoms listed here and also the scoring 0 to 4, and it creates a total symptom score of 0 to 13. Now this measure is was developed about 25 years ago before rigorous guidelines for Piero development were published. But it was not developed with patient input about the wording or the response set. The response items, as you can see here, they combined symptom frequency and symptom severity, and that could end up being confusing to participants. On the bright side, it does take less than 1 minute to complete electronically. It's been recently empirically validated, and it does look like it can detect change pre and post ERCP in patients with PSC. This last item is a 1-item numeric rating scale, the itch row, and it evaluates itch severity, and you can see a picture of it here. It's an 11-point scale. Patients complete this twice in the morning and the evening via electronic diary. It appears to have good psychometric properties based on a poster that was published or presented at EASL in 2020. I think it was initially developed to evaluate pediatric itching. And there is an observer rating form that's completed by caregivers for pediatric itching. And this was used to support a drug label claim in 2021 for a specific drug for pediatric itch. This measure is now an adult itch PRO. It's been used for 2 drug studies for itch in PSC and PBC patients, and I've listed the 3 studies here. So what I've described earlier is broad PSC concepts in the literature and all of the Piero instruments that have been used in all different kinds of studies. I wanted to focus more specifically on what are intervention or drug trials and clinical trials doing. So I did a search of PSC, adults, phase 1 to 4 clinical trials, interventional in the US, and I retrieved 39 clinical trials. Now there's about 60 trials in total, and there's also observational and patient registry data. But just for the purpose of this exercise, I just focused on these 39. So of these 39 trials, half of them are phase 1 or 2 trials, and these are small studies. They're focused on dosing and safety, and so they're really not appropriate to use the Piero measure in most of these phase 1 trials. But the other half, there were 18 studies that were phase 2 or 3 trials. They're larger and they're more focused on treatment efficacy and safety. And of these 18 studies, only 5 or 27% evaluated a Piero endpoint. So this would be a number that we would hope that we could see improving over the next several years. Of these 5 studies, one of them had a primary endpoint that was a patient-reported outcome. It was a reduction in itch related with PSC. And you can see that they used 4 Piero instruments, such as the itch row and the Pse Pro. The other 4 studies, the primary endpoint was a disease endpoint like change in ALP, but they had several secondary endpoints, and some of those were patient-reported outcomes. And we have them, you can see here, for itch and fatigue and symptoms, and they've used 4 different types of instruments here. So we are seeing more of this, and hopefully more of this in the future, putting PROs into studies, even looking at primary endpoints that are disease markers. So PROs in clinical trials is an opportunity. What we're finding is that less than 30% of these phase 2 or 3 clinical trials are using PRO measures to evaluate how patients feel or function on treatment. It's a missed opportunity to see if treatments improve biomarkers, symptoms, or both. Without PRO measurement, we're unable to capture any potentially clinically meaningful benefits to patients with how they're feeling or functioning. Recently, there's been these new Piero instruments developed for PSC. The evidence base is very difficult to track in the scientific literature, very few qualitative or content validity reports in the papers, and these tools will need more investigation and modification. So we are excited to be joining, collaborating with PSC Partners and Duke on the PSC Partners PROM project. We have 3 aims of this project. We're hoping to get started this summer. The first one is that we'll be conducting in-depth qualitative interviews with adults with PSC to really understand and better characterize symptoms and life impact. We're definitely going to publish this qualitative work. This kind of work has not been published previously in the literature. Aim 2 is to work with a stakeholder engagement group with several stakeholders to make decisions about selecting a Piero measure as is, or something that needs modification, or new development of Piero measures to evaluate 3 major symptoms associated with PSC. And aim 3 is to conduct cognitive interviews with patients to evaluate understanding and comprehension of these 3 symptom PSC measures. If we decide that we need to modify or develop Piero measures, we need to ensure content and empirical validity through this process here. Content validity really comes from the patient interviews of describing their symptoms, developing a draft of the initial measure, and then doing cognitive interviews with patients to assess their understanding. This is what I really find lacking in the literature to date. So it is this combination of input from the target population that helps us generate items for the survey and evaluation of patient understanding through cognitive interviewing. This is what provides the required evidence to support content validity. Without adequate documentation of patient input, a Piero tool's content validity is going to be highly questionable. We will use best practice guidelines such as the FDA's 4 guidances. It's a how-to cookbook for how to elicit patient experiential data and how to develop a Piero tool fit for purpose in trials. And so takeaway points for the audience. Piero measures are the best way to evaluate how patients feel and function with their disease on treatment. Less than 30% of US phase 2 and 3 drug clinical trials for adult PSCers use Piero measures as secondary and exploratory outcomes. Piero measures for clinical trials have a very high standard and must establish content and empirical validity. And the PSC PROM project is going to follow best practice guidelines, including engagement with patients to select, modify, or develop new PSC symptom measures. And by using these measures in clinical trials, we're going to be able to evaluate whether newer treatments improve symptoms and functioning in PSCers. Thank you.
Hello, and good day. My name is Bryce Reeve, and I will be continuing the discussion from Dr. Donna Evon in describing a collaboration between the University of North Carolina and Duke University with the PSC Partners Group to identify a core set of patient-reported outcome measures for use in clinical trials with PSC patients. During my short time today, I will talk about some of the attributes of what we look for in an ideal patient-reported outcome measure. I'll talk about the steps today that Dr. Donna Evans and myself will follow in terms of developing this core Piero measure for PSC, and then quickly I'll run through some sort of best practices for using these measures in clinical trials. To start off with, importantly, as you heard from Dr. Donna Evans' presentation, in PSC trials, the most important thing is we want to make sure that we're capturing outcomes that are most important to patients with PSC. And so we call these patient-centered outcomes. And typically to identify these things, as you'll hear me describe in later steps as well, is we'll actually interview PSC patients and ask them what they think is most important in terms of what they want to see in terms of treatments for PSC. Now, this is sort of an imaginary, but I have no doubt that many PSC patients will describe they'd love to have a cure for PSC or at least a disease remission. And I imagine also PSC patients would love to see reduction or removal of such symptoms such as itching, abdominal pain, fatigue, and jaundice. And if we pretend like these are the more core central type of outcomes we want to measure in clinical trials to look at treatment benefit, we have to measure each one of these potentially from different sources. So for example, for disease remission, we may be thinking about something like a biomarker, like a blood sample to look at the response to treatment. For things like jaundice, we might ask a clinician to evaluate the skin tone of the patient. But when it comes to subjective symptoms like itching, abdominal pain, and fatigue, those are things where we think a patient-reported outcome measure is the best source for capturing systematically a patient's level of their symptoms. And as you learned from Dr. Evans' presentation, patient-reported outcomes means those are information come directly from the patient and no one else. And when we refer to a measure, that is either a single question or multiple questions that help us understand what is the level of itching, the level of pain, or level of fatigue overall. So now that I've introduced you to the importance of a patient-reported outcome measure for evaluating or measuring key patient-centered outcomes in clinical trials, how do we determine what is the ideal measure for use with PSC? What the FDA deems fit for purpose of a Piero measure. And the such things we'll review is validity, reliability, interpretability, and respondent burden. Starting off with validity, the importance of the validity of the measure is ask, does this measure what it's supposed to measure? So for example, if I'm supposed to be measuring fatigue, then when I pick up my favorite fatigue questionnaire, Piero measure, I should expect to see questions related to tiredness or being worn out. There are two different key types of validity that we're particularly interested in for Piero measures. And the way we evaluate these can have a different approach. The first one is content validity, and the second one is construct validity. For content validity, this asks, to what extent does the Piero measure include the appropriate content that represents the concepts that make up that particular Piero measure? And so for an example, let's pretend we're interested in assessing the outcome or symptom of itch. And typically what we do is we'll interview patients and ask them to describe the experiences of itch. And when patients talk about it, they might describe itch in terms of how severe or intense it is. They might talk about how long it lasts or how frequently it comes back. They may talk about itch in terms of what parts of their body is itching. And then importantly also, they might talk about how itch impacts their daily functioning. Now for content validity of a Piero measure, what we're particularly interested in is to what extent does the Piero measure questionnaire capture each of these four attributes of itch. So for example, if I were to develop Bryce's new itch measure, I would expect to see particular sets of questions that measure intensity, duration, the area of the itch, and how it impacts their daily lives. So that's content validity, and typically it's evaluated qualitatively through interviews, as you'll hear me describe later. The second aspect of validity is called construct validity. And this is much more of a sort of a quantitative evaluation where we collect data from PSC patients, and we are looking to see to what extent scores on the Piero measure behave in a way that's consistent with our theory or hypotheses. So for example, again, I'm going back to the example of Bryce's new itch measure. We may ask questions of collected data that like, do scores on Bryce's new itch measure relate to scores from other established measures of itch? Or we might ask and test to see if we had a sample of patients with Pureitis, do they have higher itch scores on Bryce's new Piero measure than another sample of patients who do not have Pureitis at the time? A third question we might ask related to construct validity is if we had a known treatment, maybe a ointment or cream that removes the itch, we would expect if we collected scores on Bryce's new itch measure both before the treatment and after the treatment, we should see the scores significantly get lower. So again, these are all examples of ways we test to make sure this Piero measure is performing as we'd expect to measure in terms of its validity. Another aspect we look for in a Piero measure is the reliability of the measure. And the reliability is basically, in short, is to what extent is there presence of measurement error. We don't like error, but every measure has error, whether it's a Piero measure, a clinician measure, or any other type of measure out there. So we're trying to reduce the amount of error in the measure so we get a true expression of the true systematic differences between patients or over time when we collect that particular data. Another way we look at the quality of a Piero measure is how interpretable are the scores. And so as an example of this, let's pretend like you took Bryce's itch score measure, and it comes back and says your itch score is a 64. And probably in your mind going, well, thanks, but is that a good score, a bad score? Does that mean I have severe itch or mild itch? I don't know. So ideally, any type of appropriate Piero measure will provide some information about what might represent high scores versus low scores. High scores might represent more itch. What's the highest score? Maybe 100 is the highest score. Does the 64 represent none, mild, moderate, or severe pain? What are meaningful changes in scores that are meaningful for patients? So all these things are important for us to know to be able to make inferences about whether the patient's itch is getting better over time and how much better it is in terms of its interpretability. The last aspect we're interested in, of course, is we want to make sure, since we're asking PSC patients to provide their own time to answer these questionnaires, we want to try to reduce their burden. So having 100 questions on your itch would not be ideal. So we need to find a minimal number of questions that we can ask you, but still provide a reliable and valid estimate of their score and how it changes over time. So those important are always the key to us to make sure that we are reducing and maximizing your participation on a clinical trial. So now that I've summarized sort of the key attributes we look for in a measure, how do we actually evaluate the validity and reliability of a Piero measure? And this involves multiple steps, as you see on this particular screen. And these steps are typically consistent with what the FDA recommends, as well as other organizations and institutions. And so the first step, and I'll be going through these in more detail in subsequent slides, the first step is to figure out what we should be measuring. Those are the key patient-centered outcomes. Step 2 is identifying what's the appropriate measure. Step 3 is evaluating the content validity through qualitative interviews. Step 4 is then making sure that we have a construct valid and reliable measure using a quantitative research study. The fifth step typically is after the measure gets established, then every single study that uses that measure keeps building that body of evidence about how well that measure performs. And as I said, we have a unique collaboration of the University of North Carolina, Duke University, with PSC partners to carry out the steps you see on the screen, especially steps 1 through 4. So for step 1, how are we going to identify what are the most key important outcomes that patients with PSC want to see in terms of treatment benefit? And typically we approach this through individual interviews. Well, we'll have our trained interviewers sit down with PSC patients of diverse backgrounds, and we'll ask them questions like, please describe what it's like to live with PSC. You may also ask the question, please describe your symptoms during a flare-up. And let's pretend the PSC patient says, well, itching is one of those symptoms. Our interviewer might do some further probing with, well, how does your itching feel at its worst? And then they might probe, well, how does it feel during your day? And then how does your itching interfere with your sleep at night? And then they might also ask, how do you know when you have a treatment for itch, how do you know it's actually working? What are signs you look for? So these are kind of questions that help us understand is not only which outcomes are important to you all, but what are the attributes we want to look for to measure treatment benefit? And the second step once we know which symptoms and functional impacts we want to assess in PSC trials, then we need to figure out are there Piero measures out there that already exist that we can actually use for PSC studies? And so we'll go through a review of the published literature and then ask, is there already a measure out there that measures the symptoms that we're looking for PSC? What is the evidence for the validity and the reliability of these measures, as I outlined earlier? Has this measure actually ever been used in PSC patients and evaluated? Would this measure benefit from some modification to adding some questions, changing some questions, adding some instructions? And ultimately, if we don't find there is a good quality measure out there and we don't think we can actually make changes to it, then we might be forced to develop a new Piero measure from scratch. Once we identify which measures we'll be working with for PSC, the third step is, to what extent do we feel like these measures are both understandable to PSC patients and content valid? And again, this is a very qualitative approach using individual interviewers with a diverse sample of PSC patients. And during this process, what we'll do, because remember, now we have the Piero measure, and let's pretend like we're using Bryce's new itch scale or measure. We might show you a question from Bryce's measure, like, my itching interfered with my sleep, none of the time, some of the time, most of the time, or all the time. And our interviewers will then probe with the patient and say, when you look at this question, please tell me in your own words what this question means to you. And then after the patient describes the question in their own words and then tells us what their answer would be, that the interviewer might say, well, why did you select most of the time as your answer? And then the interviewer might probe other ways about the question and say, okay, you answered most of the time. What days were you thinking of? Were you just thinking about your sleep last night, your sleep over the past week, your sleep over the past month, or the sleep during your last flare-up? So all of these are important information that helps us understand how well the questions in this Piero measure perform and whether they're capturing the right aspect of itch or whatever symptom we want to measure. In step 4, we then ask, what is the reliability and the construct validity of the measure? And typically, this is a quantitative approach where we collect data from a much larger sample of PSC patients, and we will not only have PSC patients complete our new measure or established measure, but they might also complete other related Piero measures and information about their demographics, like socioeconomic status, their age, as well as their clinical characteristics of their PSC. And then we'll conduct statistical analyses looking at reliability of the measure and the validity of the measure in terms of association of the measure with other clinical data and Piero data, and if scores change over time consistent with our theory. Once we know that we have and have established our core set of Piero measures, then we will work with the PSC community about how to implement these measures in their regulatory trials or clinical research studies. And there is a set steps or procedures as outlined by the Proteus Consortium. You'll see the website in the bottom right of my slide there, that help guide investigators and researchers about how to integrate these established Piero measures in trials with PSC. The first question is, will the Piero be a primary, secondary, or exploratory endpoint? Now, if the focus of the trial is to cure PSC, then typically that will be the primary endpoint. Cure, it might be measured through a biomarker where the patient-reported outcome might be a secondary measure. However, if the treatment is like a cream or ointment for itch, then the patient-reported outcome might become the primary measure in that study. So that's important for us to know. In step 2, of course, we'll select the measure. In step 3, we're interested in, well, how often should we collect this patient-reported data? Should it be 1 time, 2 times, 3 times? So how often should we collect it? And should we collect it by computers, through a tablet? Should we collect it on paper or through a smartphone? In step 4, we need to set up a statistical analysis plan for how we're going to evaluate treatment benefit using the patient-reported outcome measure and who will be within our sampling plan for the clinical trial. In step 5, we're interested in how to make sure we get complete data on the Piero measure. We'll conduct the analyses appropriately with our analysis plan, and then ultimately write up and disseminate the results in a timely manner to all stakeholders, including patients. So in summary, the 3 key things I want to note is, number 1, is patient-reported outcome measures are the best source to systematically collect direct patient experiences in clinical trials. And number 2, our team at UNC and Duke University will follow best practices to develop this core set of Piero measures in PSC research. And most importantly, the last aspect is patient involvement is the most important aspect to make sure we develop a valid and reliable measure. Thank you for your time.
Thank you so much, Dr. Evon and Dr. Reeve. Dr. Korsnick? Thanks. So I'd like to introduce Dr. Reeve and Dr. Evon. Dr. Donna Evon is a clinical health psychologist who's worked in the field of chronic liver disease for over 20 years. She conducts patient-centered outcomes research and uses quantitative surveys and qualitative interviews to understand patient experiences. She's also collaborated with patients as research consultants on several of her projects. Her research has been funded by the NIH, PCORI, and industry. Dr. Bryce Reeve is a professor of population health sciences at Duke University School of Medicine, where he serves as the director of the Center for Health Measurement, trained in psychometric measures and methods. Dr. Reeve's work focuses on assessing the impact of disease and treatments on the lives of patients and their caregivers. This includes the development of patient-reported outcome measures, PROMs, using both qualitative and quantitative methods, and the integration of patient-centered data in research and healthcare delivery settings to inform decision-making. For our Q&A, I'd like to begin. There are a couple of questions that people have asked in different ways. So for the first question, they've asked things in different ways. One is looking at what tool exists for capturing the holistic impact of Pureitis on quality of life, anxiety, and depression, because it's such a common quality of life issue. And we have a second question that also asked about the potential of how many PROM aspects you all were thinking of covering. So Donna, do you want to start?
Sure, thanks, Joanne. In terms of instruments to measure Pureitis, so what we're finding being used in the clinical trials is the one like the itch row that I talked about in my either a visual analog scale or a numeric rating scale. So one item measuring basically itch severity. There is a measure called the 5D itch scale, and it measures 5 dimensions of itch, and one of them is the effect of itch on life, life interference. So with work and social life and sleep. That maybe has some merit. That was developed in several different patient groups with cholestatic disease. PSC patients were some of them, but not the full sample, and that measure would need some more work. There is another quality of life measure for Pureitis that's put out by the Pureitis Foundation in Europe. It's copyrighted, unfortunately. So I don't know, we'd have to kind of look at that. But to my knowledge, there has not been an itch quality of life measure used in many PSC studies and certainly not in clinical trials.
Oh.
With regard to the second question, the rationale for focusing on 3 major symptoms is just, I think at this point, it's kind of a financial and financial funding kind of timing issue at this point. When we do those qualitative interviews, we will be asking about all symptoms and publishing that first paper of its kind to really describe multiple symptoms. But then when we go on to do the instruments, we're going to be focusing on 3.
Thank you, Donna. A quick question for Dr. Reeve, which is, excuse me, one sec, sorry. How do you evaluate construct validity or pre-post transplant impact or construct validity in PSC when there is no drug that works and no gold standard for PROMs?
Yeah, that's an excellent question. Thank you for asking. You know, and unfortunately, for many of the measures that I've been involved in, I work across a range of chronic conditions and in pediatric and adult populations. And many of the times, either A, we don't have a gold standard to compare our measure with, like fatigue, or B, we lack some known treatment that we can use to determine if our measure is sensitive to change over time. And so because of that, you know, there's a few things I want to note. Now, number one is at the beginning, when we plan our approach to how to validate a measure, we take in consideration what are those opportunities for us to be able to validate the measure qualitatively and quantitatively. And the second key aspect is that no single study by itself is going to say this measure is valid or not valid. Typically, for any measure, you're building or accumulating a body of evidence about how well this measure performs in different types of settings and contexts of use. And through that body of evidence, give you more and more confidence about how that measure is doing. So in short, you know, if we're developing a measure, and let's again, going back to the original question, there is no treatment out there that we can look at change there. There are a number of other ways we can look at validity in its current state. One is the qualitative methods. And so again, that goes back to the cognitive interview, where we make sure that when patients read these things, these questions, that they are both understandable and relevant in a valid assessment of itch or fatigue or abdominal pain, whatever the outcome is. So that's one key aspect, the content validity. And then when it comes to the construct validity in quantitative, so again, we're trying to figure out, well, what can we use to help build this body of evidence? So if we look at change over time, what we can do is think about what, you know, known groups validity. My example in my presentation was for itch. If we knew that there was a subgroup that has extreme itch or pruritus, and another group that doesn't have itch or pruritus, and we test our scores and we find statistically significant differences, that helps provide support there. So again, it's just something, and then when treatments ultimately come on that we know work there, then we're in a better position to provide that validity evidence.
Thank you so much. There's another question that has been upvoted several times. Donna, there's been a lot of interest in using apps and other health tracking and technology to assess patients' symptoms continuously rather than just at single time points, for example, at the doctor's office. Is that a part of this project?
That's not going to play a large role in the qualitative interviews, development first draft, and kind of understanding, patient understanding of the items, response set, and et cetera. but that is definitely, I think we really need to be, with a future eye here towards digital technology and how easy it is going to be for people to be doing what we call ecological momentary assessments, where they're on your smartphone or through your email, but coming in via text. So I think that's clearly an avenue we need to take a look at when we think about how we're going to monitor and collect the data. And I think that we also need to be thinking about the timing of symptoms, and whether these are symptoms, each of these symptoms, are they things that do change on a daily basis, a weekly, a monthly, because we want to map on kind of the frequency of our tool with the way that we kind of anticipate symptoms changing.
Thank you. Here's a question. What is the best measure for fatigue and what questions are asked? Bryce?
Great question. I know there's lots of great measures of fatigue out there, and at this position there, I'd hate to say what is the best measure for PSC until, we've had a chance to review them. And when we review these measures, we're looking, we can't probably answer it now, because what we want to first do, and that's part of the project that Dr. Evon and I want to do, is we want to talk to PSC patients first about what that fatigue experience is. What's it like in terms of its severity, in terms of tiredness and worn out? How does that impact their day? So we can't answer that question until we actually hear from PSC patients describe fatigue. Then we're in a better position to make sure that whatever fatigue measure is out there has the same types of questions or items that are consistent for what we hear from PSC patients. And so what we hope to do is, after we hear about the qualitative nature of fatigue, then Donna and I will go through and select what we think may be the best measure. And then we'll actually do testing of the same measure with PSC patients and have them actually take the measure and tell us whether they think that measure is appropriate or is missing something. And so only after we do that sort of process there, we're in a position to say what's the best fatigue measure out there. Lack of anything, I have been involved in the National Institute's Patient-Reported Outcomes Measurement Information System initiative, the Promise initiative, funded by the NIH, to develop core outcome measures to use in a range of chronic conditions. Fatigue is one of the measures, including the measure. Ideally, it may be one of the things we look at, because it has been used in so many different populations. But again, I don't want to say this is the best fatigue measure until we've gone through those early steps.
Thank you so much. Donna, Dr. Evon, sorry. Please comment on steps taken to preserve patient confidentiality.
Oh, that's a great question. Just with protecting patient confidentiality in any kind of research study. So when we get started with this, this study will be approved through the IRB board here at UNC. We will collect that data, and that data stays in a confidential database. Patients are assigned kind of, a unique subject ID. That data stays just within the study team. When we go to publish it, so let's say, for example, in that qualitative paper where we're going to describe several symptoms associated with PSC, we will there's no identifiable data in that whatsoever. Even when we go to provide like a table of several, demographic or patient characteristics, we will not have data in there if the cell doesn't include like at least 3 patients in a cell. We will describe actual quotes, because these will be recorded. We will describe an actual quote that somebody provides to describe what their itch feels like or their fatigue feels like, but it won't be associated with anything identifiable. Everything will be de-identified.
Great. 2 other questions I'd like to fit in. Bryce, I'll ask you first. Does diversity contribute or not to a PRO?
I'm sorry, does what contribute to a Piero?
Diversity.
Oh, diversity. Oh, yes. Oh, thanks. Oh, I love that question. I could spend the next hour talking about that. So importantly, when we are determining what is the best measure, actually, even before that, let me even step before that. When we actually trying to figure out, the first step is what are the important outcomes for PSC patients? We need to make sure that there is a diversity of perspective, diversity in terms of both experience of the condition or disease itself, but also in terms of the population for which we're trying to generalize the results. And so you can imagine, importantly, when we conduct our qualitative interview and our quantitative methods, we need to make sure that we are including individuals from diverse backgrounds in terms of age, race, ethnicity, nationality, and so many other, gender, and so many other factors. And so importantly, as we approach each step of our process there, we actually have we put a purposeful sampling design plan where we try to make sure that we particularly target key populations to make sure they are present in the development process. We also, in addition, want to make sure that what we're developing is appropriate for all these populations, particularly when they might have different perspectives on the outcome or the measure themselves. And so again, these are things that we work routinely throughout the process to make sure that all these populations are reflected in terms of what are the outcomes and what are the appropriate measures, and are the measures written in a way to make sure it's valid for all these populations for which we're targeting our interventions.
Thanks. Dr. Evon, why is it so important to translate and validate PROMs in multiple languages?
I think that probably gets back to a little bit of what Bryce was just discussing in terms of diversity. So I'll say that the translation of an instrument is a whole other kind of process that Bryce has done several times. But so taking something in English and then translating it for another country or another language is a whole separate, additional process for validating an instrument. But it's very important if we want to develop one instrument that can be used, in several countries with several languages, but not part of what we're doing, with our 3 aims at this point.
Okay.
But when we look at measures and deciding what's the best measure, we'll be looking to see if these measures have been translated to multiple languages. Because if there's imagine if there's 2 good measures, A and B, otherwise all equal, but one has a lot of translations associated with it, that might be one that we particularly choose overall. And then the translation process includes not just the direct translation, the words are the same there, but we need to make sure the meaning is the same in both these populations. And so we'll do the translation and then do both qualitative and quantitative evaluation to make sure that we're capturing conceptually equivalent concepts across languages.
All right. Dr. Evon, do you have a quick closing thought?
I just want to say, thanks for having Bryce and I to your annual conference today, and we look forward to getting started on this project soon. And I know that there are a lot of parents and caregivers out there, and I know right now what we're focused on is a measure for adults with PSC. It needs to be kind of the first step, but also knowing that a lot of times these adult measures can then be later translated into a measure for peds, for children, or for caregivers or parents to complete, and so that can kind of be a step down the road.
Great, thank you. And so I'd like to close and say thank you so much, Dr. Reeve and Dr. Evon. After this exciting session, we are thrilled to move into the closing session with Drs. Korsnick and Pratt and Ricky Safer. Over to you, Dr. Korsnick.
Thank you very much. And thank you for that session. And delighted to say you've almost made it through. We're at the closing session. You've heard over the last 48 hours a good deal about the state of the art of PSC, what questions are being answered and what questions are being asked. You hopefully have a very extensive appreciation of both how much we're learning, but also how much we have to do, how much we still have to learn. You've heard about advances in genetics and the microbiome and the environment, and with regard to interactions of IBD with PSC. There's ongoing research in genetics, in omics, in the microbiome, and immune profiling, all of interest and seeds of hope to direct us towards therapies. We've heard about exciting efforts to understand the epigenetics of PSC, which may be a key driver of inflammation. We've heard about promising new therapies such as berberine linked to ursodeoxycholic acid, and about new imaging methodologies. We've also heard about the possible impact of nutrition on PSC. We've heard about ongoing work at both pharma and academic centers, of large efforts and work of some of the academic PSC centers, such as our own, the Resnick Center at BWH at Brigham, and in Norway's PSC Center, as well as others, and of efforts of individual researchers. Each study builds on the works of others. So I'll turn it over to Dan to discuss a bit more about what we've covered.
Thank you, Josh. Well, everyone, we've reached the end of 2 really busy days. A lot has been covered, a lot has been achieved. I wanted to start with just a brief reflection on why this organization, this conference, are so important. When my PSC patients come for their routine clinic visits, time is spent to not only address the standard medical issues related to their PSC, but equally importantly, I take the time to understand how my patients are processing and coping with their PSC diagnosis. My patients often talk about the importance of having access to education with accurate information, and the importance of being part of a community of people on a similar journey. This message was heard again in the wonderful PSC Patients Perspective session this morning, moderated by Mary Vias. This is exactly the void that PSC Partners helps to fill for some patients and their caregivers. And while that role is absolutely essential, PSC Partners aspires to and achieves much more than that through their support of PSC-related research. PSC Partners has awarded now over $5 million in research funding through their annual research grant program. PSC Partners have engaged with an accomplished group of 16 researchers and clinicians who form the Scientific Medical Advisory Committee. The involvement of these PSC leaders highlights the level of support and commitment there is in the academic community to support patients with PSC and PSC-related research efforts. It is this group, along with the PSC Partners Board of Directors, that reviews all grant applications. The importance of this seed funding cannot be overstated. Along with Dr. Alan Mullen, a hepatology colleague of mine at MGH, I was blessed to receive a PSC Partners Canada grant a few years ago that allowed us to establish an organ oil program for patients with PSC that we hope will provide important insights in PSC and produce data that will allow us to be competitive for larger grants down the line. Happily, PSC Partners remains committed to funding their annual research grants. But now let's switch to what has gone on during this conference. What we have learned these past 2 days is that PSC Partners has much bigger plans for their support of PSC research than just individual research grants. The launch of the International Collaborative Research Network was the highlight of this meeting. Generally speaking, the goal of the ICRN is simple: accelerate PSC research for the benefit of patients with PSC. How does PSC Partners hope to achieve this aspirational goal within the context of the ICRN? To start to understand this, we can look one last time to the Castleman Disease Collaborative Network, the research model we heard about yesterday, developed by Dr. David Fagenbaum and his team to accelerate research and treatment for Castleman disease. And they achieved their goals by encouraging and facilitating collaboration among the global research community, mobilizing necessary resources, and strategically investing in high-impact research. Most importantly, and just as we are doing with the ICRN, patients and their loved ones are important and valued stakeholders in the process. One important step in the ICRN process has already occurred. It occurred yesterday with the small group think tanks. It will help guide the development of the PSC Partners Strategic Research Plan. But what was exciting is that today we have heard about parts of that research plan that are already underway. Multi-year initiatives focused on supporting drug development. The first is the WIN study, the Worldwide Integration of Natural History Databases. Excuse me. This is an exciting initiative with the goal of enrolling 2,000 patients in a true natural history database. Goals include identifying novel biomarkers, developing external control arms for clinical trials, and gathering prospective quality of life data. The second is the PROM project, what we just heard about, the patient-reported outcome measure study. We know that reducing symptom burden and improving quality of life are important endpoints of clinical trials. The PSC specific PROMs are lacking. This is a project designed to fill that important need. And the final important point I'd like to make is that progress in our understanding of what causes PSC and identifying effective therapies for PSC has to have an interdisciplinary and collaborative approach. This realization is something PSC Partners understands and embraces. I particularly love Dr. Carlson's slide of the PSC clock and all of the active areas of research trying to understand pathogenesis. But what I particularly love was his point that while each of us may have a particular area of research expertise and interest, we have to be open to all possibilities if we are to succeed. I leave this conference feeling energized and very optimistic, and I hope you do too. The combination of a vibrant, talented, and engaged patient community with committed and accomplished researchers and clinicians is destined to succeed. It has been a pleasure and an honor to serve as your co-host. I will look forward to seeing you all in the near future. I'll turn it back now to Dr. Korsnick.
Thank you, Dan, and I certainly agree with all that you've said. I think the research presented here serves as important guideposts. Whether they work, whether they succeed in their initial aim or they don't, we learn from all of it. Advances are happening. All of these efforts build on each other, and none succeed in isolation. And certainly what you've heard about here is just a small sample of the extensive research that's being done throughout the world. Several of the high impact research projects that Dan highlighted are being developed by PSC partners, including the launch of the ICRN, the International Collaborative Research Network, PSC Natural History Project, WIND, as well as the PROMs, all efforts underway, which are large, impactful studies. All of these bring together a large number of patients, researchers, and others to advance the field. The PSC Partners has been the critical link to facilitate, expand, and catalyze these programs. Our discussions as part of the ICRN at this conference are helping to shape the PSC research agenda. This effort will grow in importance and will guide researchers, clinicians, pharma, and other stakeholders. The process of how to intertwine PSCers and researchers and clinicians is an ongoing effort and will take some time to refine and understand how best to make it work. The work of David Fagelman and the Castleman's Disease Collaborative Network, as Dan mentioned, is really about how this type of effort can succeed with a broad impact on therapies, and their work is pointing a way forward for our efforts. This conference has been important both to learn about advances and hear the patient voice, and ultimately to bring in the patient voice to help shape the research agenda. We're all engaged in a process which we will hopefully find a way and help us as a community identify the fastest and most direct and most certain path to therapies for PSC. Seeing this progress is tremendously exciting. For ICRN and the research agenda, these discussions are not the end. We'll continue these conversations and work together. The PSC agenda will be further refined in the months ahead. The research agenda will serve as an important guide, and together we're creating an important infrastructure for natural history data, of biobanks, and of the ICRN, with an increasing number of groups interested and helping to make these studies and programs happen. The journey may seem long and too slow, but with the growing community being gently pushed along and encouraged by PSC partners, we're making progress that's accelerating. It may sometimes be difficult to appreciate the advances when you're experiencing a disease that is moving at its own pace in your body. I understand, and we need to move ahead with a sense of urgency and impatience. With all this understood, I am tremendously optimistic and excited. Some efforts can take a while other advances can happen more quickly and unexpectedly, but they all build on each other. I anticipate several therapies for PSC will emerge in the years ahead, both to treat inflammation and to reverse fibrosis. Though it may seem implausible where we are now, I do believe that to expect a cure is a reasonable hope and expectation in the not too distant future. And now over to you, Ricky.
You're on mute. Ricky, you're on mute.
Ricky, you're on mute.
Sorry. Okay, let's start again. Thank you, Dr. Korzenik and Dr. Pratt. And Dr. Korzenik, I hope what you said at the end will happen. So we all feel this sense of urgency, and thank you for understanding. So while this has been a really long but promising 2 days for all of us, and thanks to the expert leadership from our 2 co-hosts and additional insights from our wonderful international speakers, together we've accomplished what we set out to do: to present an overview of the state of international PSC research, to champion the patient voice as we continue to break down silos and other barriers slowing research, to spark multi-stakeholder collaboration and facilitate patient-driven projects and patient-focused drug development. We ended up with 550 attendees registered this weekend, 149 of which are researchers, clinicians, or pharma. So our so-called tiny mighty army is actually expanding. And to our co-hosts, Dr. Josh Korzenik and Dr. Dan Pratt, we couldn't have dreamed of having better partners to launch our most important undertaking to date. Your trust in us and your enthusiasm in collaborating with us to turn this conference into reality fill us with deep gratitude. You welcomed us into a territory that has been traditionally exclusive to clinicians and researchers. You encouraged us when we took the driver's seat, you advised us, and tirelessly participated in hours of planning sessions. I remember 3 years ago when Dr. Korzenik made us aware of the Chan Zuckerberg Initiative RARES 1 grant and agreed to be the medical lead along with Dr. Chris Bolus. Looking back at where we were then, I realized how far we've come. And this is just the beginning. We look forward to continuing to work together in this fight with whatever it takes until a PSC diagnosis comes with a cure. The PSC Partners team and the PSC community thank you both for your dedication to hearing and honoring the patient voice. I want to go through a few highlights. First, from day one, during the presentation of the draft research agenda, 28 excellent questions were shared by the community. These questions reflect the PSC patient voice and guide us as we push forward the most important research projects on the quest for a cure. 148 attendees participated in the ICR network breakout rooms yesterday. The most popular rooms were cause of PSC and genetics and inflammation and diet and nutrition. The topics of greatest interest were the cause of PSC, the PSC-IBD connection, and inflammation. Discussion of environmental factors, diet, and nutrition were also of great interest to attendees. Community engagement in the outstanding poster abstract session was high, with more than 30 questions posed to the presenters. And then a few highlights from today. In the patient session at the beginning of the day, we heard the open and honest stories from members of our PSC patient community about their journeys with PSC. There is no one-size-fits-all journey with PSC, and we should all lean on each other to help us get through the tough times. Research updates today, Dr. Green taught us about what is known about the cause of fibrosis in PSC and shared data from several outstanding posters about efforts to treat it. Dr. Carlson shared the incredible work being pursued in Oslo related to the genetics of PSC, the use of bile duct on a chip technologies to study PSC, and much more. Dr. Korsnick taught us about the connection between IBD and PSC, especially focusing on the role of the gut microbiome. There's lots of exciting research going on at his center and others to better understand the connections between PSC and IBD. In the Natural History and WIN think tank, natural history studies allow us to learn about the factors that predict PSC progression, identify biomarkers, create external control arms for clinical trials, and more. The WIND initiative will bring together many centers from across North America and Europe to gather high quality and standardized data from 2,000 patients over 5 years. There was quite a bit of discussion about diversity, both in terms of patient demographics and the characteristics of their disease. Diversity is so important to ensure that a natural history study is representative and valid. The WIND initiative will ensure that a diverse patient population is included. And in the last session, we learned about the importance of an effective fit-for-purpose patient-reported outcome measure in clinical trials so that PSC symptoms and quality of life are captured. Thank you as attendees for joining our think tanks to discuss the programs that PSC is launching within our ICR network to fill the huge gaps in PSC research. There is amazing power when patients and caregivers work closely with the scientific community to unite and harmonize international efforts to accelerate research and drug development. This weekend, we've heard the words hope, excitement, collaboration, progress. This is just the start of the ICR network. As we further expand the network, we will need continued input and guidance from all stakeholders. We will formulate a formal strategic research plan to guide our research efforts and expand our grants program that already supports over $5.5 million in international research grants. We will focus on supporting drug development through our 2-year multi-year projects, WIND and a PSC-specific PROM. Our PSC community is truly blessed by the number of passionate clinicians and researchers who are dedicating themselves professionally to improve our lives and provide us hope for the future. Our Scientific Medical Advisory Committee will continue to lead us, and we can't thank all of you enough. We also sincerely thank our industry partners who are running clinical trials to discover new therapies to lighten our disease burden and to find that elusive cure. And the patients and caregivers in our community are at the center of our ICR network. So everyone, please come along with us on our journey together. I want to finish up with just a few reminders about upcoming community programs. To continue our educational sessions from July through December, we will hold monthly post-event webinars covering everything from coping strategies, nutrition, clinical trials, symptom management, liver transplant, and beyond. Be sure to join us for these informative webinars. And still to come this afternoon, the exhibit booths are open from 2 to 3 p.m. The time to celebrate session for our patient community to unwind by connecting with new and old PSC friends will start in a couple minutes. When I finish, please go back to the platform and click on the Time to Celebrate tile. Patients and caregivers, please join us tomorrow morning starting at 9 a.m. Mountain Time. You get 1 extra hour of sleep. For our peer group sessions, our chance to share concerns, questions, and encouragement with our PSC peers who understand each other so well. If this is your first conference, you really don't want to miss tomorrow. So I'd like to end with one more huge thank you to absolutely everyone who has helped us make this conference and our inaugural ICR network so successful. Oh, the places we can go. Together, we're creating a world where a PSC diagnosis comes with a cure. Thank you, everybody.